In this module, we're again going to be using the genome browser, which you access by clicking on the genome browser on the left panel of the starting screen, and then you are brought to the starting genome browser page. Now, for this module, we're going to be looking at DNA activity of specific regions. And in order to do this, we're going to make use of one of the tracks that we have available called the Epigenome Roadmap. Now, the Epigenome Roadmap consists of many different profiles that we have compiled that look for DNA activity based on histone modifications and chip data from these profiles. So as you can see, there are many different profiles done in different cell types looking at different histone modifications. And we have then put together a summary of these modifications that you can view on your genome or region of interest. So we've activated that, and now we're going to click Redraw. And here you now see that you get your region of interest. In this case, we're looking at the region of the MYC-N gene with the epigenome roadmap presented below. So just as a brief overview, the different colors represent different functional annotations of the genome. So if you have white or gray representing the majority of the area within this chart, it indicates regions of silence or not a lot of transcriptional activity. If you have regions that are red or green, this would indicate transcriptional activity, red specifically indicating transcriptional start sites for the most part, and green indicating active transcription and transcripts being transcribed, and yellow representing then regions that would represent more enhancer-like functions. So you can see here that we're currently looking at the MYC-N gene. For this example, we are going to have a look at the TERT gene, just to illustrate how we can use this epigenome roadmap to understand DNA activity in certain regions. So now we see that we are at the TERT gene, and again you see that you do have some red signal at the transcriptional start site, and you do have low level of transcriptional activity in some of the profiles. But for the most part, the majority of the TERT gene is actually white or gray, indicating that it is not being transcribed across the profiles. If we zoom out a little bit in order to get a broader view of these areas, so you can see some of the genes that lie adjacent to TERT, here you can see where there are genes that are heavily actively transcribed as opposed to the regions of no transcription between the genes, and then a lower level of transcription across the TERT locus. Now, as I've said before, this is a summary of all of the different profiles. So these are the different profiles laid on top of each other to give you a cumulative illustration of the signal across the different profiles. If, however, you want to look in a more detailed view and see the individual profiles in different cell types, you can, of course, go down to the epigenome roadmap again, and here on the toolbar next to it, you can see that you have different modus or different ways of looking at this representation. And if you click Plugin instead and Redraw, you will now generate a more detailed view of the profiles that we were summarizing before. So now each specific profile has been split into its own row, and they are now grouped based on cell type. So what you can see here again is that you do have, for the most part, under the TERT gene, generally inactive regions of DNA. So as you can see, the graph is interactive. So if you hover over, you get an indication of what category it is. So here you see that it's quiescent or low activity, and also a label of the type of tissue that it's in. For instance, here where you see that there is some active transcription, you can see that it is at an active transcription start site, and this is in peripheral blood cells. And here even you do have, in a subset of profiles, active transcription across the TERT gene, where you can see that it actually is occurring in iPS cells or in human embryonal stem cells. So there it's expected that you actually would have active transcription of both the transcription start site and the transcript itself. If we zoom out a little bit, just to further demonstrate the difference between genes that are only partially active in certain profiles and those that are generally expressed in a broader overview of profiles, here you can see that the genes that lie next to the TERT gene here you can see that they are actively transcribed basically unanimously across all different profiles that we have. Now we've shown you how to look at all the profiles at once, both in a summary form and in the individual profiles with one per row. However, maybe you want to select individual profiles or a subset of profiles to look at to make it a little bit less messy. And that is possible by going back to the transcript view. One way you can do it, if you want to select an individual profile that you might already know that you want to look at, here you have all the different profiles you can select from, so you might want to look at this specific profile in the IPS cells. You can select that and simply click redraw, and here you will get the one profile below your region of interest. If, on the other hand, you want to look at a subset of multiple profiles, that is also possible. Here, what you simply select is custom, and then you scroll down to the bottom, and here you can see that you can have a custom ID, and here you can either enter the IDs of profiles if you happen to know what they are, or you can click on edit, and here you get a list of the different profiles that are there, and you can simply click on the profiles that you wish to include. So we'll just select a few examples here, and we then drag them over into the active profiles and confirm, and then we're going to redraw, and here you see that we've specifically selected a subset of the profiles that are now shown for our region of interest. So one additional piece of information that should be mentioned about looking at these types of profiles is that they are visible when you're zoomed in onto specific regions of the DNA. However, if you do zoom out too far, so we'll zoom out another 5 times just as an example, as soon as you reach a threshold of above 200 base pairs per pixel, you're then not going to be able to visualize these profiles anymore. However, as you can see here, the transcript views, the epigenome roadmap track is still active. So if you were then to zoom back in on a region, the profiles are still there. So now we've looked at an example of how you can use the epigenome roadmap in order to look at activity of DNA and the different types of functionality of different regions of DNA. However, you might want to look at which transcription factors might be involved in regulating the DNA at these specific regions, for instance, at a transcription start site. So in the next module, we will be looking at using transcription factor binding site profiles. So if you're interested in looking at that, please watch the module on transcription factor binding.